Okay, then I would like to welcome you all to this around 1 hour webinar where we talk about Gene Set Enrichment Analysis, how to analyze different types of data, proteomics, RNAseq, and single cell. And as you know, this is a demo today, not a hands-on exercise, and I will send out the slides after this session. I think most of you have attended the basic training recently, or maybe another training sessions, or you are an existing customer. So hopefully you already know how to handle the software, because I won't get into detail regarding that in this webinar. So I assume that you know that you have some experience with the software. And my name is still Ann Holmberg, and I've been with Qlucore for 10 years now, actually, so time flies. And I would like to apologize for all the confusing messages you got regarding how to join this webinar. This is another webinar service that I normally use. But because there are so many attendants nowadays, I thought we need to use a more powerful tool so that we get good performance in the webinar. So I'm still learning how to do this in the best way. So I tried it out on you. Sorry for that. But I think most of you actually managed to follow the all the instructions and do the right thing, which in the end turned out to be register on the homepage. Okay, this is the agenda for today. So we will start talking about gene set enrichment analysis, a sort of pathway analysis. This will also be a hands-on exercise in the tomorrow's advanced training, which you can also register for on the homepage, that you actually need to register for on the homepage. I will send out an email regarding that as well. Then we will have a look at how to import and analyze proteomics data. And the same with RNA-seq data, BAM files, how do we import that? And we end today's session with an example on how you can work with single cell data. So I will show each part, and then I will open up the chat and so that you can ask questions. But while I'm presenting, I will probably not see your questions. So the first thing that we will talk about is Gene Set Enrichment Analysis, GSEA. And in this, you match a gene list from your data set with public gene sets. And the gene sets is a compilation of lists, typically from pathways or other biological contexts. And so typical data repositories for where you can find the gene sets is MSigDB, the Molecular Signature Database from Broad Institute, Reactome database, the Gene Ontology Consortium, KEGS, and so on. That's where you can find gene sets. And it does not need to be genes. The gene set enrichment analysis is basically just a tool for comparing lists. So if you have good pathways for proteins or metabolites, for instance, you can use them as well. So in this exercise, we are going to find enriched pathways for genes that are good at separating T-cell leukemia, TALL, from other types of leukemia. We are going to use a public leukemia dataset, and we are going to use a pathways or a gene set from Reactome. And then once we have found interesting pathways, we are going to have a closer look at them by use them for visualization in synchronized plots in Qlucore. So a sample plot and a variable PCA plot, and that will give us more information about the pathways that we have found. And there is a lot of more info regarding this in the help in the software help menu, documentation and help manager. The how-to guides use GSEA. So let's have a look at this. So I will open up the software. Here we are. And I will load the dataset, and this is one of the built-in example dataset that you will find in the help menu. And I take the big one, acute lymphoblastic leukemia. Here we are. And here is the sample PCA where we see the 132 samples. And down here we can see that we have 22,000 variables. In this case, they are probe sets. And just to show you what we are interested in, I color this on leukemia subtype. There are 7 different subtypes of leukemia that the patients have been diagnosed with. And what I'm interested in now is the genes that are good at separating the T-cell leukemia, T-ALL, from the rest of the samples. And as you can see, it's easy to find thousands of genes that are very good at separating this pink, orange group here from the rest of the samples. So I'm interested in finding pathways where these genes are enriched. So I actually don't need to do what I did right now. I don't need to start by doing the statistical analysis at all. This is only to show you which genes I'm interested in. What I need to do now before I do the gene set enrichment analysis is to make sure that the unique identifier in my data set is same as in the pathways that I'm going to compare with. And in this case, the pathways that I compare with, they have gene symbol as their unique identifier, whereas in this data set, as you can see, I have probe set ID as the unique identifier, because this is an Affymetrix data set. Yeah. So the first thing then I need to do is to make sure that I also use gene symbol. And gene symbol is one of the, it's present in the data set as a variable annotation. So I can just change the unique ID here, the identifier, to gene symbol. And then, as you can see, if I go back here, we have 13,000 unique genes, but we still have 22,000 data points. And that's because there are several probes per gene. So I need to collapse, meaning that I need to create one value per gene, and I can do that in a number of ways. Either pick the maximum, the minimum, the average, or the median. So let's say that I create an average where there are more than one value per gene. So now the data set was recalculated, so now I only have one value per gene. If you work with RNAseq data, for instance, you will already have the gene symbol as the unique identifiers. You probably don't need to do that. Sometimes, though, there are duplicates, and then the gene set enrichment analysis will complain, and then you will still need to collapse your data set. Okay, so this is the preparations that I need to do. So then I open the Gene Set Enrichment Analysis workbench by pressing on this little launch button here. And that opens up this window here. And here we can see what we have, the name of the dataset, how many samples, how many variables, and that we have the right identifier here. And here I have the gene sets that are available for comparison. These are examples that are shipped with the software. We don't update them, so you should not use them. Instead, you should download the gene sets that you are interested in using from one of the data repositories that I mentioned. And then you put them somewhere on your computer in a folder, and then you just point to that folder here, and then your gene sets will show up here. And please note that the gene symbol is case sensitive, so you cannot compare human with mice, for instance. You must make sure that you have the correct gene symbol, otherwise it won't find anything. You can also create your own GMT files. This is a very simple text file format, and it's described in our reference manual. So you can just search for GSEA and GMT and you will find what it looks like. Okay, so that's the first thing I need to do. I need to select which gene set or gene sets that I want to compare with. So I select the Reactome pathways subset that we have downloaded from the Reactome homepage. And here we have more than 1,000 human pathways in this. Then I select the metric because I'm using all of my genes here. I have not pre-selected the best ones. I'm using the full list of genes, but I will rank that list so that it mimics what I'm interested in. And I'm interested in genes that are good at separating TALL from the rest of the samples. So then I would want to rank according to the T-statistica, to the so the two-group comparison is what I want to rank by. And that means that my gene list is sorted so that I have the best upregulated genes in the beginning of the list and the best downregulated genes in the bottom of the list. Okay, then I press run. And this takes a little while. So now each of the genes in my list is compared with each of the 1,000 lists, pathways, and this is permuted 1,000 times in order to calculate the Q value. And in settings here, you can decide how many permutations you want. You can even increase this number. And so here we have the results. So this is actually very fast. And here we see the results. So cholesterol biosynthesis comes up here with a good enrichment score and a good Q value. And here we see the running enrichment score. You can see that we have a positive maximum. And this value here equals that value there, the enrichment score. Each blue line here is a matching gene. So we can see that we have a lot of matching genes in the beginning of the list, because this is the list. Here's the beginning of the list, here's the end of the list. So this tells me that I have a number of upregulated genes here that are enriched. If we go down here to the bottom of the list, you see that we find a couple of good negative enrichment scores. Here, for instance, we have interferon gamma signaling. And here we can see that we have a negative maximum and a lot of downregulated genes that are enriched here. Let's sort on Q value. And let's filter on Q value. So let's say that we can accept a Q value below 0.15. The Q values here are generally a bit higher than in your normal statistical analysis. As you remember, the Q value is the false discovery rate. So you can accept Q values up to 0.2 or 0.25 and it's still okay. So here we have now found 3 lists that are pathways that are really good. For each of them, you have the running enrichment score. You have a heat map showing how these genes are expressed in your data set, and a detailed list of all the matching genes, whether or not they are enriched like that. If we mark all our 3 lists of interest, we will also see that we have a leading edge overlap here, telling you if there is a big overlap between the lists. And in this case, you see that no, it's not. It's only overlapping with itself. That's good. If I now would like to export all of this information, I just press results here and decide where I want the results. That will export and create all of this information. So here we have all the enrichment scores. Here we have all the heat maps, including the leading edge overlap, and so on. So you just need to press one button to export everything. If I press lists, what happened then is that I export these 3 pathways back to my normal working environment. So let's go back. Okay, so here we are now again. Here is the sample PCA plot. Let's open up a new synchronized plot. And make that a variable PCA plot. Okay, so now here we have all the 22,000 variables. Here we have the 132 samples. So let's filter down to the best genes again. So I filter down to the 700 best genes or something. And then you see that the genes here, they divide up in 2 groups. Meaning of that is that genes over here in the same position as the samples over here, they are upregulated for these samples. Genes over here in the opposite position of these samples, they are downregulated for those samples and upregulated for some of these samples. And I can show you that by coloring the variables according to how they are expressed for the TALL group. So I just use the color mouse tool and do like that. And then you see that all of these genes are red, meaning that they have a high value for what we have colored by, the TALL. And all of these are blue, meaning that they have a low value. Now we are going to use the pathways that we have exported over here. So now I want to see which of these genes that belong to the cholesterol biosynthesis pathway. You see that there are some yellow ones hiding here in the back in the among the upregulated genes. Let's use only the cholesterol biosynthesis list as input. Then you see that I find 5 genes here among my best genes that belong to the cholesterol biosynthesis. If I want to know which ones they are, I just use the label tool. And then I see, okay, interesting. If I would like to save these 5 genes, I can just make a copy of the active list and say that these are genes cholesterol biosynthesis separating TALL, like that. And then maybe I would like to have a look at the interferon gamma signaling. So let's include everything again and color according to interferon gamma signaling. Then you see that we get a lot of pink genes over here among the downregulated genes. That makes sense because here we had a negative enrichment score. So we expect to find downregulated genes. We also find one upregulated gene here. And again, if I would like to save that, I just, if I want to see which ones they are, I have them here. If I want to save that again, I just make a copy of the active list. These are interferon gamma signaling separating TALL. So you see that there is a seamless interface between the Gene Set Enrichment Analysis and our normal working environment. So you can actually get a lot of more information regarding the pathways that you have found to see which genes are the most relevant. And of course, which genes that show up here depends on the filtration that you did initially. If you want to include more genes, you could say, I want maybe start with the 1600 best genes, then of course you find many more. Okay, so that was what I was going to show you here. So I now open the chat. So let's see. The question was, where do you see that you have 700 genes? And I see that down here. That's where I see how many genes that are active down here or in the active variable list here. Another question is, what is a good enrichment score? So let's go back to, I think you should focus more on the actual Q value than the enrichment score, because the enrichment score is really just the height of the curve. But of course, the higher the enrichment score, the more clear peak you have. So I think it's hard. So it's the normalized enrichment score that you should focus on. So based on this, my guess would be at least 1.5 or so. But I think you should Google on that or try to find some more information on that, because I don't have any real expertise on this. And then the second question is, if I have two groups which I want to compare, control and treatment, which group should I select in the two-group comparison? It doesn't really matter. The one that you select is where we will, when we talk about upregulation, then it will be upregulated for this group. If you do it the other way around, if you would choose treatment here, you will find upregulation for treatment. If you choose control, the upregulated ones will be upregulated for control. So it's only what is considered upregulated and downregulated, but the genes that you find will be the same. The pathways that you find will be the same. I hope that was clear. Any other questions? Okay, then we continue. Oh, here we have a, can we call the gene size shown in the GSEA table in the hit size? Can we show the gene size? So the matches is actually the hit size, if that's what you are asking for. This is the size of the list. This is the number of matches. So all the genes in the list is not present in my data set. So, okay, great. Good. Then we continue. So now I close the chat again. Now we're going to talk a little bit about proteomics. So I will give you a background to the data set, and then I will show you how to import a TXT file using the wizard. We will pre-filter and do a log transformation, and then we will do a visualization and analysis. And I will also show you how to eliminate batch effects or covariates in this data set. So this specific data set is a comparison between dying and surviving cancer cells. And normally, so when you do chemotherapy, that will cause detachment and death of your cancer cells. And normally when you study the effects of chemotherapy, you only look at the attached cells and skip the detached cells. In this study, you want to test the hypothesis that the detached cells actually also contains interesting information. So the experimental design is that you have 90 samples, 90 cells, 45 attached and 45 detached from 3 different cell lines. They are equally divided between attached and detached. And then you have 3 drugs plus control that are also equally distributed between the cell lines. And this is a mass spec data set max quant from the PRIDE archive, and it has been the study has been done by Karolinska Institutet in Stockholm. So that I got one more question on gene set enrichment analysis, but I will take that together with the questions on proteomics. Okay, so let's have a look at this then. Okay, maybe while I still have the GSEA open, I will have a look at this question that I got on that. So the question is, the results shown include the expression value of the individual genes, right? So it's not just a present-absent representation of the genes, because we compared two groups. I'm not sure that I understand this question. Can you please send that to me in an email, and we can try to figure out more about the question, and then I can give a good answer to this. So please send me an email after the session. Okay, so here we are. So let's just now close down this data set. And a good way of closing down data set is to go to the log tab and just either close down one specific plot or the full data set like that. And let's also make sure that we have closed the gene set enrichment analysis like that. Okay, so now we will load the proteomics data set. So I do file open or open with wizard. I find my data set, I have it here. This is a TXT file that we open with the wizard. First question is still, is this a raw count matrix? It's not. I will show you how to work with a raw count matrix when we come to the RNAseq. Right now, no, this is not a raw count matrix. It's a proteomics data set, which is the separator. It's tab separated. If you have an older version, it will say /t. Rows are variables. Yes, that's correct. We have the protein IDs here. The top left cell of the data matrix, this is the top left cell. And we go select top right cell. We go all the way to the right. You see there are a lot of zeros in the data set. We can already see that. Variable annotation header cells. This is a cell describing the variables. Sample annotation header cells. Here we have them. Or I can press shift and go to the last one, like that. Finish. That will bring the data set in. So here we have our 90 samples and 5,600 proteins. Okay. Let's have a look at the data. I go to the table view here. And I see two things. I see that there are a lot of zeros here. And this data is not log transformed, because when it's log transformed, you would see values between plus minus 15 or so. Okay, so I want to do a pre-filtration. I want to take away proteins which has mostly zeros in them. So let's start by that. So I go here to data, pre-filter, put it on, and then I go in and specify. So I want to remove variables that have a value of less than something low, 0.1, in say 80, 70, or 80% of the samples. So before we had 5,600, now we have 3,700. So I took away 1,900 proteins here. And then I would like to log transform as well. And as you can see, the log is not available. And that's because I have a lot of zeros and I cannot log transform when I have zeros. So I need to threshold again to something lower than any other value in the data set. So let's choose 0.1 again, like that. Now I can log transform. So now you see here. Okay, so some of the values are really, really high. So I actually have values over 30 here. So Good. Then I go back and have a look at the PCA plot. Okay, so this is how it looks now. So these two steps are really good. Normally it's very good to log transform your data, and if you have a lot of missing values or zeros in your data set, it's good to get rid of some of those variables. Okay, so let's try to understand what it is that we see here. So we have the annotations here. So what we are very interested in is, of course, the attached and detached, and it looks like there is a big difference between them. The blue ones are the attached and the yellow ones are the detached. We can also see that there seems to be bigger differences within the detached group than in the attached group. We have more annotations. Let's open up a new synchronized plot and use more colors here. So we have 3 different cell lines. We can see that they are equally distributed between the attached and detached. We can also see that there are big differences between the cell lines. Let's have a look at the drug. There are 3 drugs and a control. Let's color on that. And we can also see that there are differences between the treatments. We also have another annotation called set, which I'm not sure what it is. This could be batch, for instance. And that's also a strong signal in this data set. Okay, but so when we want to analyze that, say that I would like to analyze the difference between detached and attached. The way to do that is to do a t-test on source and choose either attached or detached here, and then just drag the statistical slider to find a number of genes that are good at separating the attached from the detached. And we see that there is a big difference with them. We saw that immediately when we pulled up this data set. If we feel that we have covariates in this data set, for instance, we might have a big batch effect, then we might want to take that away. So if we think that set is a batch effect, we can take that away. And we can even take away the effect of cell, if it could be that we are not really interested in the differences between the cells, or we are not even very interested in the differences between the drugs. We are just very interested in the differences between attached and detached. So that could be one thing. So the eliminated factors, which we will talk more about in the advanced training tomorrow, is a way of taking away covariates in your data set. It's very important that the covariates are equally distributed between the groups that you are looking at. Otherwise, you risk taking away a signal that you are actually interested in. So we can have a look at the result here in a heat map. So here we are. Here we have a heat map of the remaining 1,300 genes. Maybe we want to go a bit lower. We might want to add a fold change filter as well. So maybe I want to filter on a fold change 2. And I go down to the 500 best genes. Let's view the whole heat map. Let's order it hierarchically clustered. Like that. Let's color on source, for instance. Okay, then we see that, yeah, we can see that there is a big difference between the attached and detached. We see we have one sample here that is over there. We can actually use the flip then and try to move that. That's in this cluster, like that. I got a question on the scale of the heat map. When we read in the data set, we normalize it to mean 0, variance 1, putting all the variables on the same scale. That normally is what works best with the PCA and the heat map. You can put that back, but then normally the heat map gets much it's harder to interpret the heat map. So this is normally a very good setting for both the heat map and the variable PCA plot. So that's why we have the +2 -2 scale. Okay, in the heat map, as you know, we can also show more colors. So if I would like to include all the other ones, the cell, the drug, the set, for instance, I can do that as well. Like that. Okay, let's go back here again. Let's say that now instead I'm more interested in looking at only the detached cells. So I take away the attached for the moment, and then I would be interested in, let's just do like that so that we are back here. And then maybe I'm interested in the difference between the different drugs. Like that. And so then we can do that. So then I do a multi-group comparison, an F-test on the different drugs to see if I can find interesting signals here. A little bit of a mixed picture, but we definitely see differences here. And then again, we can visualize them in the heat map. And as you know, the heat map, let's take away everything here. And let's color instead on drug. And you know the heat map automatically updates when you do something, the active genes will automatically show in the heat map. And here we have the situation that the heat map, when we do the clustering, it's not that clear. And then, of course, you can choose to say that I don't want the hierarchical clustering. I actually want to decide how to sort the heat map. So then you can say that I want to sort on drug. And then it could be that the heat map is more easily interpreted. But then, of course, it's not unbiased anymore. Good. So that was a few examples on how to import and what you can do. And if we go on here, more options with proteomics data is that you often have individual results files. And as long as they have the same layout, not necessarily the same proteins, that's not a requirement, but the same layout, then we can use the wizard to import all of these files. You just select all your files, and then the software will create the data matrix for you, leaving with a lot of holes in it, of course, because there will be proteins that are present in some samples and not in others. And you will find more information in the help documentation and help manager, getting started, import data and annotations, protein and proteomics. So there is more information on this. So let's open the chat and see what we have here. So yeah, so there was a comment that set could refer to experiment replicate. Maybe there was an annotation called replicates as well. So yeah, we can just guess what it means. Yeah, exactly. So in this last analysis, I didn't remove the covariates, which of course I could have done or should have done. So I just wanted to show you different options that you can actually focus on only the detached cells if you want. But of course, you could have done it in the same way. You could have removed the batch effects here as well, because they were equally distributed within the groups of the detached as well. More questions? No, then we move on. How to search for a specific protein or gene. So if you are here, for instance, you can say that I would like to understand where this one is situated in my heat map. And I can use the mark. and that will highlight that, or it should. So here so let's just see. Mark. At least that's how it should work. Let's do like that. Mark. Oh, I don't know why it doesn't do that. So let's see. Let's active. Let's zoom in. That. We can order the variables on. Yeah, I only have one thing to order by, so I'm sorry. I can order here I can order on the protein ID, so I get them in alphabetical order, and on a number of other different things, or I can order hierarchically clustered. And I don't know why this isn't giving me. This is what I expect, that this should actually help me find that specific one. I must have a closer look at that. I would have expected that that mark would actually mark that in the. Yeah, I'm not sure why this isn't working, so I must have a closer look at that. Or if you want to search for something specific in the list, you can just quickly here type. So if I want to, if I type an F, it will jump to the F and so on. And you also have a search possibility here. So here you can say, I want to search for a specific protein. Okay,